Welcome to this video in the Blueprint of Life topic. This video is going to be looking at the dot point, discuss the potential impact of the use of reproduction technologies on the genetic diversity of species using a named plant and a named animal example that have been genetically altered. So we've been looking at two examples, being a plant example of BT cotton and an animal example of salmon with human growth hormone that has been inserted into it. So in this video, we're going to mostly look at the impact overall of reproductive technologies on genetic diversity of species. And at the end, we'll tie back in those plant and animal examples that we need to, but knowing that we've discussed them quite a bit in class. So within a population, we know that variation occurs naturally. So it has to occur naturally, as this is helpful as environments are able to change. Organisms that possess the variations that suit those changes are able to survive, and the organisms that do not have the characteristics that suit those new environments obviously don't and therefore die. So we need variation in the population so that those organisms are able to pass on those characteristics, and therefore through Darwin's theory of natural selection, survival of the fittest means that those that are able to deal with the changes in the environment reach reproductive age and pass those characteristics on to their offspring. Reproductive technologies that we have looked at include cloning, artificial insemination, artificial pollination, and transgenic species. Now, with all of these different reproductive technologies, in most cases, the genetic diversity of a population will decline with the introduction of any of these reproductive technologies. So, when reproduction is allowed to take place naturally, we have the processes of meiosis, we have the processes of independent assortment, random selection or random recombination of alleles during the process of sexual reproduction. So all of those things increase our genetic diversity. However, when we're pinpicking exactly what characteristics we want to pass on from generation to generation, obviously we're selecting the same alleles each time. Therefore, that genetic diversity is going to decrease. So from this diagram, we can see that each population has a gene pool. When reproductive technology is introduced, whether it's cloning, transgenics, artificial selection, or artificial insemination or artificial pollination, the number of each type of allele available within that population will change, and it will most likely decline. So if we have a look at the example here, if we decide that the brown coat for cows is favorable, big B, and we kill off all the other cows, so those containing the little b, whether they're heterozygous or homozygous recessive, we will effectively be removing all little b alleles from the gene pool. This means that all future cows will be homozygous dominant, only carrying the big B genotype. This leads to obviously much less genetic diversity in this population, as every single cow is going to have exactly the same genotype and therefore going to pass on exactly the same alleles. So again, if we had a environmental change, so something to impact on brown fur could be that the temperature increases considerably and those darker animals suffer more from the heat, then obviously we're going to see a gradual decline in the overall population because they're unable to adapt to their new surroundings. So the reason why genetic diversity declines with cloning is that we are carrying out asexual reproduction and basically producing identical offspring. So we've seen with bacteria produce asexually. We start with one bacterial cell, divides to become two become four become eight, and all 8 of those bacterial cells are exactly the same as the original parent cell and exactly the same as each other. That's why we see when we introduce antibiotics, we're able to get rid of bacteria quite quickly. However, because they do reproduce so quickly, there can be slight variations amongst the population as well. But we get rid of majority of those bacterial cells in one hit because they are all exactly the same. With artificial insemination and artificial pollination, we are choosing the traits we want and ignoring all others. So basically saying, these are the alleles we want and put them together. Remember that the traits that may be favorable for us may not be favorable for animals or the plants that we are changing. For example, we are breeding much larger chickens, so we have more meat, but this is causing pain and suffering for the chickens as their legs are not growing bigger in relation to their body. And as we can see in this diagram here, in the 1950s, this first chicken is about 60 odd days old, whereas the second chicken here is nowhere near that age. But as you can see, it is considerably bigger, and that is because of the introduction of things like human growth hormone, which we've seen in the salmon that we've talked about as well. Transgenic species initially increase the genetic diversity because we're creating a new gene. So we're inserting the gene from one organism into another. But after a while, the genetic diversity will decrease again because we are only then producing that one type of animal. So they all have that new gene with the desired trait inserted. Okay, also, it could also be one type of plant. Okay, so transgenics can be used in both plant and animal species. So this slide here just shows us the overall impact. So we've got our two examples in the middle there. We've got our plant at the top being Bt cotton, our animal at the bottom being our human growth hormone for the salmon. So Bt cotton produces its own pesticides and therefore is pest resistant. So a chemical is produced in the leaves that stop insects from being able to eat it. They also produce higher yields of cotton at lower costs. However, over time, resistance may occur as the insects that are eating the leaves may develop a resistance, and then the chemical that it produces is no longer effective. With the salmon, we can see genetically modified salmon at the back, which is much larger in size in comparison to the same age salmon that is grown in a salmon farm. So they grow to a much larger rate, a much larger size, much quicker, which obviously improves the economical value and food production because we're able to produce greater salmon meat, which we know is quite expensive, also has quite a lot of health benefits for people, and also reduce pollution because we're not adding chemicals to the water in order for the salmon to survive. So looking at the overall impacts, on the yellow arrow we've got our positives, where we initially increase biodiversity as it introduces new characteristics. Okay, there's a potential to increase the gene pool by reviving extinct organisms such as the Tasmanian tiger. So this although this actually hasn't happened yet, it is a possibility with these new genetic technologies that they're looking at. Negatives include loss of genetic diversity, entire populations being susceptible to disease because of that loss of genetic diversity and lack of variation within the population, the lack of overall biodiversity. So if we have a lack of genetic diversity, organisms start to die off, then obviously we have a lack of overall biodiversity because we're losing individual species. And lastly, risk of contamination between genetically modified and unmodified species when they're released into the wild. So people are really concerned that we don't actually know what impacts on our health that genetically modified food and genetically modified products could have. So there's still a lot of concern about whether or not there are going to be health impacts or impacts on the environment when these genetically modified species are released out into the wild. And that brings us to the end of this video. So thank you for watching.